Hi, everyone, and welcome to today's webinar, The Lab of the Present, Agentic AI in Action. I'm happy to see all of you here today. My name is Liz, and I'm this webinar moderator. I'm also a senior marketing manager at Quantori, and I'm excited to guide you through this session. We have an interesting journey ahead of us exploring AI-driven R&D. Let's take a look at our agenda today. First, we will have a brief Quantori intro. Then we will explore the industry perspective on AI-driven R&D. We will cover expert systems models and then dive into a live demo of QScientist and Q models. Finally, we're going to wrap up with interactive Q&A. For housekeeping, please keep your questions in the Q&A section here in Zoom. At the end, we will answer them and you will see which questions were answered. Before we start, we would love to learn more about you. Let me launch a short questionnaire. Just a moment. So you should see it on your screen. Let's take a minute to see the answers. Okay, now I see people start answering the questions. That's great. Only 4 people replied. Come on, it's anonymous. You don't have to be shy. Okay, we have already some statistics here. Only 8 people. Okay. Just a couple more seconds and I will stop the survey. Okay, let's see what we have here. So most of the people came here from email invitations. Nice. People are working mostly in technology, AI, and data science. And people would like to learn more about the agentic AI. Okay, that's great. Thank you everyone for answering the questions. And let's get back to the presentations. Okay, let me introduce our amazing headline. We have here today Chris Waller, Quantori's Chief Strategist. We have a guest, Tudor Apreya, CEO of Expert Systems, and Alisa Vershinina, Senior Scientist and Business Analyst. And without further ado, let me pass a virtual microphone to Chris. Chris.
Super. Thank you, Lisa. Let's go to the next slide. And I've got great news for everyone on the call today, on the webinar today, who came to see Agentic AI. You're in the right place. So if you haven't heard about Quantori, it's probably not a little surprising. We're a small company, about 6, 7 years old. We're a professional services firm that services the pharmaceutical and life sciences industry. We are pleased to claim 10 plus of the largest pharmaceutical companies as our clients and working with a number of smaller companies as well. We are globally distributed as a services organization and currently on staff. You'll see we're 500 plus engineers and scientists, and that is one of our differentiating factors, is that on our engineering team, we embed PhD and master's level scientists that understand life sciences and pharmaceutical research and development. And you'll see that we partner with almost every hyperscaler on the planet. So next slide, please. It is important, as I mentioned earlier, we do bring specific domain expertise into the projects that we work on for our clients. And I will not drain this slide, but I just wanted to give you an indication of the areas where we tend to focus and see most of our work being oriented. So pharmaceutical R&D, clinical research and informatics, a large practice around scientific and laboratory informatics, and then some work in the regulatory compliance spaces. So we have a broad portfolio of offerings across most of the early R&D value chain in life sciences. Next slide, please. I've been at Quantori now as a chief strategist for 4 or 5 months, and I've been working on our position relative to the challenges that I see in the environment related to wet labs and dry labs. And we talk about lab of the future. We've been talking about lab of the future for a long time at the Pistoia Alliance and other places where I've tend to work and interact. What I'm trying to take us to today is a pivot. Let's pivot away from discussions around the lab of the future and talk about the realities in our wet and dry labs today and move into a lab of the present. And so if you think about in the wet lab and the dry lab, really, there are challenges, fundamental challenges related to data fragmentation, the fact that scientists spend a huge amount of their time working on repetitive tasks and dealing with data. The automation itself is rigid, and this notion of the black box, so a lack of data being born fair, really. And in the dry lab, very similar, right? Model staleness. So thinking about your predictive models that you invest in going stale immediately when new data are generated. The notion of lack of computationally assisted hypothesis testing, and then the harsh reality of the in silico designs failing to provide developable results. And then lastly, and we all deal with this every day, is the technical debt. The fact that we've invested so much in our custom work and scripts and IT infrastructure in general that it's a reproducible tax on every project. So we have a lot of barriers to overcome. And so what we'll talk about today is, can we use agentic AI and AI in general to help us address many of these issues? So let's go to the next slide, please. Before we jump into that, and this is a little bit of a busy slide, I did want to ground us a little bit on some of the terminology that we'll use today. One of my pet peeves is using AI to mean everything that has a computer attached to it. So we're not going to use that as a definition today, but we will talk a lot about agents. And in the purest sense of an AI agent, it's an autonomous system that perceives, makes decisions, plans, reasons, and achieves a goal, often using a lot of different tools or services. So think about an agent as an actual replacement for a human persona in a laboratory setting doing something. An AI workflow is a step-by-step process that can use smaller agents to accomplish larger tasks, right? And often those workflows depend on agents that are tapping into APIs or models in a sequential or parallel kind of fashion. And then lastly, an AI service is what we all know and love as typical services that are accessible through APIs, and those services typically do a specific task. And these things work together, right? Agents will use services to accomplish a task. A workflow uses services to accomplish a larger task. And then these agentic workflows combine both structured and agent-like agents to uh, accomplish often human in the loop type workflows. And that's what we'll talk about today when we get into the demo. We'll be using agentic workflows with a human in the loop as to accomplish these tasks in the laboratory setting. So let's go to the next slide. And I've got an image of a slide that helps explain a little bit the vision that I have around what an agentic laboratory might look like. So this is a nano banana generated diagram that I really like. It shows up as my background in a number of other presentations. But in a typical wet lab, dry lab scenario, we think about things like the DMTA cycle, the design, make, test, analyze cycle. And if you put that into an agentic framework, you can think about the personas of the scientists that you need that are performing these types of functions. And in this particular case, I've called out a number of personas. There's an architect agent that works in the very beginning of the stages to do the design. There's a conductor agent that's helping make the compounds. There's an observer that's making sure that the tests are going the way that they're, or, the way that we expect. And then there's an oracle that pulls it all together at the end to look to see just exactly how well the experiment went, and then helps the architect agent start all over again, right? So in an agentic, purely agentic setting, you could imagine executing your DMTA cycle using agents based on these personas, all orchestrated through a platform and constrained by a knowledge graph. And what we're talking a lot about these days is, in the context of agentic workflows and LLMs more generally, is using knowledge graphs on top of and around the corpus of data that we're using and the LLM is based on, to actually ensure that the results of the LLMs and the work that the agent does falls within a specific domain or a set of truth that we all believe. So, given that as a vision for an agentic lab, let's go to the next slide. What we've done at Quantori specifically to address the agentic, I guess, the emergence of agentic workflows and platforms in the life sciences and pharmaceutical area, is to come up with a set of solution accelerators that are pieces of code or know-how that allow us to get to solutions for our clients faster. And you'll see on this slide that they are, in fact, zero gravity. We use these accelerators to build bespoke solutions for our clients, and they become our clients' IP. By design, they're extensible, customizable. By design, they're multi-cloud, but can be cloud specific, and can be made to optimize particular cloud environments. They're obviously life sciences domain savvy. And if you think about the tools that we've gone to market with, or at least talking about mostly in the market, are tools like Q Scientist, which we'll talk about today, and you'll see a demo, which is our flagship offering in the agentic space. And Q Scientist is accompanied by a number of other tools. The other one you'll hear about today is Q Models, and you'll see Q Scientist working in concert with Q Models to perform design type functions within a laboratory. So I don't want to drain this slide too much, but there are a set of infrastructure solution accelerators as well as life sciences specific solution accelerators. And we're constantly updating this set with new Q Suite offerings, which you can find on our website. Next slide, please. And at this point, I'll turn it back to Lisa.
How is everyone feeling? Thank you, Chris, for the intro. Now we're excited to give the floor to our partners at Expert System in just a moment. But first, before we do, we would love to learn a little bit more about you. Let me launch one more quick questionnaire. This time we're curious to know which AI agents you are using. Okay, let me launch it. Should be on your screens. I will give it about a minute to get your answers. It's only 2 questions now, so please don't be shy. Let's give it 30 more seconds. Oh, people are more active now. That is nice. Thank you, everyone. You're making one more marketer happy. Okay, 10 more seconds. Okay, let's see what we have here. So the first question was, which AI platforms do you use? And people are either using commercial closed source AI platforms or not using AI platforms yet. Okay, I see. And do you know how to integrate AI models into drug discovery workflow? Most of the answers we got were conceptually or not yet. I haven't shared them. You can see it for yourself. These are our results. And thank you for your answers. And without further ado, I'm passing the virtual microphone to Tudor. Tudor, the floor is yours.
Yes, hello everyone. Thank you, Lisa and Chris, for giving me this opportunity to discuss some of the machine learning models that we have been building at Expert Systems. Since Quantori is busy building the lab of the future and artificial intelligence-based systems like Agentic AI, I just wanted to make sure that everyone in the audience knows that there is a difference between machine learning and artificial intelligence. So very briefly, with artificial intelligence, you have a system that mimics human cognition, and you pretty much can interact with the system. Think of the ways you interact with Alexa or self-driving cars, or perhaps in the future, we will have lab scientists doing work for us. The difference with machine learning, of course, is that it's a subset of AI where you essentially have a dedicated data set, which is the training set. You have a test set on which you try your models. And of course, you have an algorithm or several algorithms that ask a question. So for instance, you can build a system that will be trained to discriminate images of cats from images of dogs. If you were to ask that question, that machine learning system, what is the molecular weight of propranolol, it would not be able to answer. If you were to deal with an AI system where you say, hey, I have pictures of cats and dogs, can you discriminate? It probably does a good job, certainly with the most modern AI systems that we've seen developed by Anthropic and OpenAI, etc. On the other hand, you have to wonder, will it be able to deal with specific questions? And the answer is probably yes. If you ask what's the molecular weight of propranolol, it will give you the correct answer. So back to the context of expert systems, what we have been building for the past 3 years is a set of machine learning models that are based on public literature, patent data, and proprietary data that we have acquired from ChemDiv and several other companies along the way. And the way we use that data is to try to ask the question, if you have a small molecule in the SMILES format, if you submit it to the system, it will try to guess whether that molecule binds to a specific target. So in total, we have about 6,000 plus targets, of which more than 5,000 are of mammalian origin, and about half of these are human targets. Considering that the human proteome has about 20,000 proteins and about 3,000 of them have been addressed with chemical matter and approved drugs, that is not a bad tally. Of course, such system would not work on all the other targets. When it comes to the quality of these, we prefer to have 2 log units spread and at least 80 compounds in the training set. Ideally, we would want hundreds of compounds in the training set. That is not always the case, particularly with newer targets. And we use multitask learning to try to address lower quality models. So that would be more of a similarity type question. Last but not least, we also look at cytotoxicity, and we have data for cell lines where we ask the question, will this compound kill this particular cell? Why is this important? It is important because if you have a compound that you particularly try to use in cancer, and it does a wonderful job on your target, having this cytotox model tell you that it's going to kill the cell anyway might indicate that this molecule does not work on the target you think it does. Of course, the ideal experiment would be to run the knockout, remove the target, see that your compound doesn't do anything to the cell, and then you're safe and you are free to pursue target engagement. Next slide, please. The other aspect that we have been critically interested in is the area called as pharmacokinetics and toxicity, so absorption, distribution, metabolism, excretion. And starting with absorption, of course, we try to ask critical questions about the small molecule properties like solubility, permeability and bioavailability. And then going through distribution, we look at fraction unbound or plasma protein binding and the ability to cross the blood-brain barrier. And of course, related to that, the fraction of drug unbound that is active in the brain. We also look at volume of distribution. With respect to metabolism, we have a suite of models, about 50 different enzymes that looks simultaneously at phase 1 and phase 2 drug metabolizing enzymes. This is important because we don't think of metabolism as a separate phase 1 and phase 2. In fact, if a drug gets into the body, there's no such thing as phase 1, phase 2. These are more human concepts, but they've been useful for educational purposes. We blend this into a single model. We also look at hepatocyte and microsomal stability. That is an important question that next to solubility and permeability would decide whether we want to progress this compound further. With respect to excretion and toxicity, we have a number of parameters that we look at. Just to give you a glimpse, I already talked about cytotoxicity. With cardiac toxicity, we have about 11 different targets that we evaluate at the same time. So we try to ask the question, does this compound, is it likely to cause QT prolongation? And in addition to hERG, there are other proteins that can be perturbed and lead to that cardiac toxicity issue. So on to the next slide. The other thing that we have developed, which we think is unique to expert systems, is a way to evaluate what is the likelihood of the compound to accumulate in a specific cell compartment, such as lysosome or nucleus or a plasma membrane. Different tissues. So it's important to think about this type of distribution in the context of the target and the disease you're trying to address. So for example, if you're interested in neurodegeneration, so working on Parkinson's or Alzheimer's disease, and then you will ask the question, does my drug get into the brain? So the question of blood-brain barrier permeability comes to mind. Efflux transporters will come to mind because you want to make sure that your compound is not actively being removed from the brain. But equally important is the target that you're interested in present in the brain. So you have to look at expression. You have to look at the capability of the compound to get into that target. So these kind of properties are important, and we try to address that upfront with machine learning. Last but not least, we also look at these properties when we do library design and chemical library optimization. So we have specifically focused libraries that would be brain permeable, or if it's a hem-onc project, we would try to maximize solubility and accumulation in red blood cells. And with that, I thank you for your attention and turn it over to Lisa. Thank you.
Thank you, Tudor.
Over to Alisa. Let me stop sharing.
Now to the demo of the platform. You see my screen okay?
Yes, it's visible. Thank you.
So welcome to QScientist, overview of the platform. What you see on the left-hand side is the variety of control and variety of systems that we have. And whenever you log in, you see your user information, you see the chats that you were interacting with before. And on the left-hand side, what I'm showing now are a variety of those different workflows. And workflows are pre-built automatic multi-step processes. We have a library of those workflows for life sciences, for healthcare, and for business purposes as well. And just setting the expectations for folks is that this is still a work in progress. So if you're interested in working with with workflows like this, please contact us after the meeting. And now moving on to the library of agents and AI models on the left, we also have this variety of solutions that we can modify, use, and adapt to specific needs. And today I will be focusing on expert systems solution, but we have library right now, and we're developing this library of different agents and different workflows and different models that we're currently integrating into to pursue a variety of tasks. Now, for the expert systems, exactly like Chris and Tudor were talking about, we can interact with the system in natural language, having human in the loop, and I'll show you how we do that here, but also I will show you what are the other capabilities and how we can really build multi-agentic workflow on a back end, and so that instead of just interacting with the chat, you will have an analysis running within the system. Now, what I'm doing here is in the chat dialog, I'm just, is anytime when you come in, I like to ask the system itself, well, what can you do? And the prototype that we have right now can accomplish a lot of those cool things that Tudor was talking about. And what I want to describe here is that the environment that you see here is the chat, and there is also another part of the system on the right-hand side where you see what we call the state graph and logs. So each time when you are interacting with the system, you're not only interacting with it in a natural language, but you also are able to see what is going on in the back end. And as a scientist or someone who doesn't maybe wants to know exactly what's going on and what might go wrong or correct, you can see what's going on the right-hand side. And for the logs, whenever you are doing any kind of analysis, we automatically keep track of what's going on, and so that whenever you have, for example, regulators coming over and asking you what happened with the particular analysis, a particular date, you will have that all written in the form of those log artifacts. Now, an example of questions that can be asked, it of course depends on you and your specific task. And like Tudor was talking about, one of the interesting aspects of this is that you can compare toxicity of different compounds. And here, rather than asking, you know, a generic question or tell me more information about this and that, with those models, you can actually perform analysis. And you can take the results of this analysis and supply them further to more agents and to answer more questions that you might have. And I also will show you in a bit how it looks like on the back end. And here I just asked the system to compare toxicity of caffeine and ibuprofen, and it's going to tell you in the end that they're kind of comparable. And here I used just names of the molecules themselves, but ultimately, of course, you might need to supply the smiles strings instead of the names, and this is also possible. And you have a variety of, you have a multiple numbers here because there are multiple endpoints for that model assesses when it calculates toxicity. Now, moving on to what else I was going to show here. I think that's it. I think I'm going to move on next to the a little bit more of information about the state graph. So here, what happens is, in addition to the expert systems head node that was thinking about your task, we have other models and other tools connected to it. So in a state graph, you're able to see that in each particular case, when you're asking system to do something, you can trace what node, which tool, and which agent was working on your task. So here, there is a variety of models that expert systems provide, but I was specifically asking about toxicity. So the system and the orchestrator agent decided that cardiotoxicity is going to be the model that is going to run in order to provide you those numbers and to answer this question. So you can track what exactly happens on the right-hand side, and you can track which tools were active. Now, here what I'm doing is I'm calling the designer. So this is the model that allows you to improve, check, and change the agentic system. And what you see here is the variety of models that expert systems provide and very straightforward agentic architecture. And what you see on the left hand side is variety of elements that can be a part of the workflow or part of the agent. Aspects of the menu, such as deployment of the agentic system and check of controls over this deployment. And what you see on the right-hand side are instructions. Let me maybe go back a second. What you see on the right-hand side are instructions. So each agent will have its name, its description, and instructions, and then is any whenever you're building agent, you want this agent to have a specific persona, you want this agent to have specific instructions, and this is all you can do in the natural language, and you can track how it looks like, and you can change it if the system is doing something that you don't like. Now, this is how it looks like inside. The agent has an instruction or step-by-step it's doing particular things. Now, this, of course, is very straightforward. So this is might not be exactly what you want, but you can improve the system. So first of all, when it comes to models and tools themselves, so the blue thing is the agent and the yellow thing is a tool. Now, the tools are pieces of code or API calls that are also can be controlled by either you or your computational team. And just an overview of how it looks like for different models. And in order to make it a better agentic system, you of course can ask it to do more things. And here's an example of where I do some drag and drops. And what I want to do is I want to create an orchestrator that is gonna, it's something similar to what Chris was explaining in his Nana Banana schematic, is that we need to have an agent that is gonna perform the tasks and decide what happens. So you can create this right in the designer. And then the next thing that I'm going to do is I'm going to add a decision maker, because when I'm running those models and when I'm interacting with the expert system solutions, I want to know, okay, I now know those different toxicities. I need to compare them and make a decision. So if I run multiple models, I have a lot of decisions to make. I have some ranking, and I can create this decision maker to do those decisions for me. And here I'm just creating a simple persona. Let's say it's a chemical informatics specialist, and provide some instructions of, your goal is to provide a go or no-go solution. If you see that your parameters are not ideal, this is not a small molecule that you want to pursue, you want to do something about it. So the agent will do a decision for it. We have a magic button that is also powered by LLM that is going to help you make those instructions better. And now I'm going to make this closed loop. So I'm going to connect the orchestrator with the decision maker and with the expert systems. Now, okay, so if your decision maker agent is telling you this, this is not a good toxicity result and you want to do something else, this is very, it's a very, how to say, it's a kind of the demonstration of what other possibilities are there. So you can connect a molecule generator, or you can connect some code, or you connect something else that allows you to keep your analysis moving forward and allows you to do something on the basis of decisions that have already been made by the orchestrator. And let's say it's some molecular generator that you connect in there. And if you decide that, no, this is not going to go well, you need to iterate more and create more molecules for your tests. And I think that, and this, ultimately you would want to create this kind of loop where decisions are made while you're running this analysis. We also have a library of some internal solutions as well. So for example, here I connected the PubMed search here. And if you want to get some published information about what you're doing, you can also do that. But the world is your oyster. Well, it's all up to you what kind of things that you want to do there. Okay, so I think that this is pretty much the end of my demo. The last thing that we do here after we designed the system is we need to deploy it and validate it. And we also have an environment that allows you to do that. And after, I'm not going to click it here for the demo, but after you save it, you can immediately go into QScientist and start testing it and start seeing what's going on and whether it works as you plan. And I think that would be it. Thank you for watching, and I'm going to pass the baton back to Chris.
Super. Every time I see that, I get excited because I get more ideas about things, agents I want to build or problems I want to solve. And I think what was stressed today, and thank you so much for Tudor and Eliza giving you the scientific and then the scientific and technical solutions for how you might overcome some of the challenges that you find in your wet and dry lab environments. You'll see the way that we're positioning agentic AI really is an efficiency play, right? We're creating agents persona based that are doing tasks that humans do. We're not currently positioning these agents as human replacements. We're positioning them as companions in the laboratory, dry or wet, to help the scientists do their work, right? And so if you remember some of the challenges that we saw in the introductory materials, there were challenges related to repetitive tasks and automation of manual workflows. Lisa showed us how to overcome that, right? We can automate those workflows. We can create agents that do meaningful things for us. And then we saw some challenges related to bringing predictive sciences into the laboratory and making those, taking greater advantage of those computational models that we do have. And we saw, the wide variety of models that Tudor and Expert Systems have created and how we've wrapped them up in something called Q models or the Expert Systems agent. And it was pretty easy, right? So that was pretty powerful. I think it was interesting to see the results of the survey around what folks are currently using as their agentic frameworks, and then also how far along on the journey are they with respect to incorporating agents and agentic workflow into their lives, right? And what I hope you got today was that using QScientist as a framework, provided by Quantori, might accelerate you on this journey, right? It's a framework that you saw that was very easy to build the agents, very easy to build the workflows. I say that tongue in cheek because it does require great skill that you saw Lisa has, but you can learn these things, right? And so QScientist as a platform is available to you to accelerate you on your journey there. I wanted to point out a couple of other things or a few other things in our suite of Q suite accelerators that might be important to you as well. Obviously, Q models, we saw that the expert systems integration. We do have a more focused effort in the life sciences early discovery space called Q Discover. So it's standard drug discovery workflows, less agentic and just more workflow driven. And then there are a few tools that I've been really excited to advance thinking around how we might use agentic solutions in more traditional spaces in the infrastructure areas, basically. So things like high performance computing. Can you use agentic workflows to manage those high performance computing workloads and jobs that we typically do in those environments? And the answer is yes. And we have an offering in that space that we'd be happy to talk to you about. LQ Portal is a interface and way to manage access and control of the LLMs that are used by a organization. And then we won't talk about Q image today, but then Q data is another agentic workflow driven solution based largely on the infrastructure in Q scientist that provides a human in the loop way of identifying, normalizing, standardizing, aggregating data. So think about that whole data curation, harmonization pipeline, and the kinds of personas that you need to typically do that. We can automate that through our Q data set of offerings. And that's what I wanted to leave you with. But the other thing I'll leave you with, which I thought was fascinating, I hope you picked up in the demo, was the magic button, right? When you're creating these agents and you're creating these workflows, the fact that we're using LLMs underneath to help create the instructions for the agents, it's an interesting evolution, that we're on right now, and I'm excited to see these technological advances in this field. And I hope that you enjoyed the webinar today, and I look forward to some questions during the question and answer period. Thank you.
Thank you, Chris. I want to remind everyone that we have Q&A section here. You can post your questions there or in the chat if you want. As of now, I don't see any questions, but I have a comment after that was posted during the demo from Ruslan. It says, "Great tool." That is amazing. Okay, let's give it a couple of minutes. Maybe people need to think what they want to ask. Okay, we have first question. Do you have a benchmark to compare AI models in the life sciences domain?
I think that's a two-door question.
Well, that's a very general question. I would say it's hard to have benchmarks when the benchmark is strictly a set of approved drugs. So assuming we would have enough data to reproduce key decisions that were taken during the lifetime of a project, which takes between, on average, 7 to 15 years before it hits the clinic, then yes, in principle, you could say one can do benchmarks and try to address this. But the short answer is it's very difficult to have such benchmarks without considering that the AI has already ingested that knowledge in one form or another. So it's rather hard.
Thank you. We have more question. What does the client need to have internally to get started on Q Scientist? How much internal effort is required on part of the client?
Lisa, are you aware of the technical requirements to make Q Scientist work? I'm seeing blank stares. I'll give you my best swing at this. Right, we got Q Scientist up and running in a few accounts right now, and it's running in a cloud environment. Right, you're going to need some sort of a cloud ecosystem to be able to house this thing. But then it generally is, it's not necessarily a /install kind of a situation, but it is reasonably easy to move it's all like Dockerized and whatever, move all that stuff over and get it up and running. So it's minimal at this point, but you do require some sort of a compute ecosystem, right?
Thank you. The next question. What is the QA strategy to ensure that the agents are doing what is expected and providing answers that are trusted? What can be represented by the system overall quality?
So I'm going to take a swing at that one. But I'm going to put a little pressure on Elisa too, just to talk about some of the agents and the performance of the agents. But if you remember my diagram earlier, I had the knowledge graph that was there that was providing the guardrails for the solution, right? So the intention is to come into an environment, understand what the clients are doing, the context of the work that they're working on, build those knowledge graphs on the corpus of data that they have, provide the guardrails around the truth, right? So you can imagine the knowledge graph actually holding the rules for how science needs to be executed within a particular domain, and then making when the agents themselves actually operate using that knowledge graph to constrain what they do so that they're doing things according to the rules and what you expect as truth is how these things are intended to operate. Lisa, do you have an expansion on that?
Yes, I can add that when in terms of agent design, each agent has a statement of roles, statement of responsibilities, and degree of autonomy. And this is something that you need to think about before you are getting into implementation of those, because you don't want them to do the things that you don't want them to do. And you need to think about what those things are when you are designing them. But ultimately, first of all, there are specific like check marks that we should follow when we're designing them. One and two, in any multi-agentic system, we can also create those safeguards internally where we, for example, if you have, um, if you are, I like this example a lot because I was working with FDA, but if you have regulators, and you think that you're in talks with FDA, you can have an agent which role will be specifically to check the work of others using also a set of rules. So you can create those back loops when the system checks itself, and when it gets to the point that it cannot decide on its own, it has you to ask questions as well and to clarify. So all of those things are elements of the design and elements of the philosophy of how you build the architecture and instructions of the agents. I hope that answers the question.
I want to add to that
Mhm.
if that's okay. On the quality, of course, from a machine learning perspective, we try to be very cognizant of the situation that machine learning can sometimes just output the wrong answers, which, of course, in an AI LLM world would be called a hallucination. So what we try to do is implement tools like distance to model and applicability domain, as well as uncertainty quantification. And we actually have all three. And when Elisa was showing the example from ibuprofen and running through expert systems, you could see in the brackets that there was a prediction and then the UQ number next to it. And of course, the lower the UQ, the more confidence you have in the model. Most of our models are multiple, so that we're running ensemble models. It's not a single model. Typically 10 models that we run concurrently. And then that range of predictions gives you an idea of something similar to, say, experimental error. And then, of course, on top of that, you can extract a similarity to the nearest molecule in your training set, as well as ask the question, how far is it in model space from your molecule? So that inherently is a way to try to answer whether the quality of the prediction is useful or not.
Thank you. While we wait for more questions, let me tell everyone that we will be happy to see you at the events we are participating soon in. The closest one would be Lab of the Future in Boston on March 2nd and 3rd. Our booth is number 25, and Chris Waller will be a panelist in the automated lab track. And on May 19th through 21st, we're going to be at BioIT World with booth number 505, and Chris Waller will be presenting again on May 20 in the AI for Drug Discovery and Development track. And we have one more question. The rule sets if then else that you type in constrain agents. Is there a library of those tasks? I would think that a lot of users would have similar repetitive setups.
It's an interesting question. Maybe I can start answering that and folks can pick up. I think that in our case, the variety of use cases that we work on right now in Quantori is very wide. So it is impossible to make a library like that, I would think, because each particular client is unique and they have their particular task that they want to accomplish. But maybe for something like ADMET process or something that expert systems are working on, it might be possible. I don't know if you, Chris or Tudor, want to answer more.
What do you think, Tudor?
Well, first I want to say hi to Chris Ho. But on the if then else, we don't have specific bins like that, but I've been working with LLMs since pretty much March 2023. And we try to log things that work in markdown format, and we try to keep those prompts or the things that we thought were successful. We try to save those so that when we run a similar task, we just reload that file. So we have ways to address this, but they are task specific. They're not yet, I wouldn't say they're generalizable to a complete set of rules that would automate drug discovery completely. However, if you say, find me patents that discuss MDCK permeability, and then find me patents that have MDCK permeability that have experimental values for MDCK, and then 5 patents that have those two as well as chemical structures associated with MDCK permeability, I think we can discuss about ways that we can do that agentically.
Thank you, Tudor. Okay. I think we are ready to wrap up. Thank you everyone for joining today. It was very insightful, I hope, for everyone. And here are our contact details. You can find 2 QR codes, or just you can use a link to access our website, or you can contact us directly at contact@quantori.com. I want to also thank all of our speakers, and especially Tudor, for joining as our guest. It was really nice to having you today. And I think that's it. If other speakers don't have anything to add, we can just wrap up.
I think we're good. I think the only thing I would add would be, the whole point of the talk today was to get you to join us on this journey from this discussion and dialogue around lab of the future as a conceptual thing to the actual lab of the present, and how we can use these tools to drive our future visions into reality. So thank you for your attendance today. I really appreciated the dialogue. Thank you, Tudor, my dear friend. And thank you, Elisa, for the wonderful presentation and overview. And thank you all for attending.
Thank you, everyone. Goodbye.
Bye.
Bye, everyone.